Hello and welcome to Basic Medical Sciences. So in this video, we're going to talk about digestion of carbohydrates and absorption, right? We will also talk about glucose transporters, right? So like in this scheme, you can see like the GIT from the oral cavity, pharynx, esophagus, and stomach. That's this first part. And then this second part is from duodenum down to the entire enteron or small intestines, right? Okay, so let's begin, right? So I told you about this slice of bread, right? So we are going to break it down into, we want glucose, pure glucose in the blood at the end, right? So firstly, you eat this slice of bread, right? So you chew that's your mechanical digestion, and as you mix with saliva, right, using the tongue, right? So in saliva, we have a specific enzyme for uh, digestion of starch, right? But what did I say? I said in starch you have amylose and amylopectin, right? So amylose is this one, a single chain, right? And amylopectin with branches, right? So the bonds on amylose are alpha 1,4 glycosidic bonds, and on amylopectin here you have alpha 1,4 glycosidic bond, right? But here and here are alpha 1,6 glycosidic bonds, right? Right. So salivary amylase, right? Salivary amylase is the enzyme is found in saliva from the salivary glands. If you remember them, like submandibular, sublingual, or parotid gland, right? So this amylase, salivary amylase, known as ptyalin, will break down these linkages, right? Alpha 1,4 and alpha 1,6, right? Like this, right? So at the end of the day, we will have like uh oligosaccharides, right? Or small carbon molecules, right? Okay, so let's go down a little bit further, because this is what happens in oral cavity, pharynx, esophagus, and stomach, right? So as like these small carbohydrates or oligosaccharides enter the duodenum, they stimulate the eye cells to produce a hormone known as cholecystokinin, right? So cholecystokinin will come to the sphincter of Oddi, right? It's found on the descending part of the duodenum at the ampulla of Vater, that's the hepatopancreatic ampulla, right? So what will happen is the pancreas will release like a lot of enzymes, right? But here we're talking about carbohydrate only, right? So here we'll only mention pancreatic amylase, right? Pancreatic amylase, what does it do? It also hydrolyzes these bonds, alpha 1,4 and alpha 1,6, right? Like this, right? So I said, like as a result, for digestion of starch, usually they just mention maltose, right? But I just want to tell you other oligosaccharides, right? So firstly, I will tell you about lactose, right? Lactose is made up of glucose and galactose, right? That's the first one. The second one is maltose, right? Maltose is glucose and glucose, two glucose molecules, right? And this one is maltotriose, right, with three glucose molecules, right? And alpha limit dextrins, right? Alpha limit dextrin is still have like some 1,6 glycosidic bonds, right? So that's where you are here. And also there is another one which is very important, sucrose. I didn't indicate it here, right? But you will see it, right? You will see it on, if we talk like on the next part, right? So far we are in the lumen of the GIT, right? So the small intestines have a villi and microvilli, which increases the surface area for absorption, right? So, or on microvilli, they have brush border enzymes, right? So the first brush border enzyme is called lactase, right? Lactase, right? The other one is maltase, and the other two, this one is more like a complex. Also maltase and sucrase, right? It's like a complex, right? So lactase, you know what it does. It digests or breaks down lactose to two molecules of glucose, right? That's lactase. Maltase, it breaks down like maltose and other like maltotriose into molecules of glucose, right? So I said lactase, right? Lactase, two molecules of glucose. That is wrong, because I said lactose is made up of glucose and galactose, right? So it gives us glucose and galactose, right? That is perfect. And then on maltose, two molecules of glucose, right? And then another thing here on alpha limit dextrans, it is digested by this one, also maltase, and sucrose is digested by sucrase, right? So sucrose is made up of two things, that's fructose and glucose, right? So the sucrase will cut this alpha 1,6 glycosidic bond, leaving fructose and glucose, right? On the on isomaltase here, there is a breakdown of alpha 1,4 glycosidic bond, and the one I indicated here is alpha 1,6 glycosidic bond, right? Which is here, right? So this will end up giving us two molecules of glucose, right? So this is in the lumen of the small intestine. We have fructose, glucose, and galactose, those three, right? So we want to take them into the enteron, right? The enterocyte, the cell of the small intestine, right? So how do we take it, right? So there is a specific channel, right? There's a specific channel for glucose and galactose, right? This one is called like sodium glucose transporter, right? Because it also transport like sodium, right? So sodium together with glucose and galactose, right? It's called sodium glucose transporter, right? That's glucose and galactose. What about fructose, right? So fructose is actually transported by GLUT5. That's glucose transporter 5, right? Now like these three, glucose, galactose, and fructose, are now in the enterocyte. We want them in the blood, right? So they are taken from these cells into the blood vessels through another transporters, right? These transporters are known as GLUT2, glucose 2 transporters, right? So the transporters which I mentioned so far, they are insulin independent. They have nothing to do with insulin except GLUT4. We will talk about it later, but for now, just to know that these glucose transporters have nothing to do with insulin, right? Okay, so now this blood vessel is actually a hepatic portal vein. So where are we going? We are going to the liver, right? So here in the liver, that's where you have your glucose, galactose, and sucrose, right? Thank you so much. If you like this video, please make sure you subscribe so that you won't miss any of our latest videos, right? So the other thing, you can take a screenshot, or you can go to the beginning of the video, take a screenshot, you draw everything, and then you can fill in the gaps, right? I think that's how you can master this. I think it's very easy, right? Thank you so much. Until next time.